Welcome to another lecture by MedicoMedics, Genetics Chapter 12, Cancer Genetics. In this lecture, we will talk about cancer genetics. What is it? Talk about different types, key genes involved, differentiate between hereditary and sporadic cancer, mention some examples, talk about some risk factors, talk about the future of cancer genetics, and end with a summary. Now, cancer genetics is the study of genetic mutations and alterations that contribute to cancer. We know that cancer arises from uncontrolled cell growth due to genetic changes, and both inherited and acquired genetic mutations can contribute to this. Its importance, therefore, is to help identify risk factors and guide the development of targeted therapies. So in cancer genetics, we are studying how changes in our DNA can cause cells to grow uncontrollably, leading to tumors. Some types of genetic changes in cancers. Well, first, mutations. These are changes in the DNA sequence, and they could be point mutations, insertions, or deletions. If you look at previous lectures, we've mentioned these in detail. Another thing could be chromosomal alterations, so deletions, duplications, translocations, like Philadelphia chromosome in leukemia, also talked about in previous lectures. And of course, epigenetic changes, so altered gene expression without changes in DNA sequence, for example, DNA methylation. Some key genes involved in cancer. So we have something called oncogenes. These genes are called oncogenes because they promote cell growth and division. Mutations in these cause them to be overactive, for example, HER2 in breast cancer. We have tumor suppressor genes. These are genes that prevent uncontrolled growth. And mutations can deactivate these tumor suppressor genes. Examples of these suppressor genes are TP53, BRCA1, BRCA2, etc. DNA repair genes. These help in repairing and fixing damaged DNA. And mutations here increase genetic instability. Examples are MLH1 and MSH2 in Lynch syndrome. So all in all, some genes act like gas pedals in a car, like oncogenes. You have tumor suppressor genes that act like car brakes, and you have repair tools. So cancer typically happens when these different systems fail. Either one fails here and this one becomes overactive, or this one doesn't work and the reparation doesn't work, etc., etc. So it's a combination of these key factors. Now, hereditary cancer is caused by inherited genetic mutations. It accounts for approximately 5 to 10% of all cancers. Example, BRCA1 and 2 mutations in breast and ovarian cancer. Then we have sporadic cancer caused by acquired mutations during life. This accounts for approximately 90 to 95% of all cancers. And the factor here are environmental exposures like smoking and UV radiation, etc. So hereditary cancers are passed down in families, while sporadic happen due to life choices or exposures, etc., etc. Now then, let's mention some examples of hereditary cancer syndrome. So we have BRCA-related breast and ovarian cancer, where we have mutations in the BRCA1 and 2 gene, increasing the risk. This follows a autosomal dominant inheritance pattern. Then we have Lynch syndrome, mutations in the DNA repair genes, like MLH1 and MSH2, and this increases risk of colorectal and endometrial cancer. We have familial adenomatous polypses. This is a mutation has occurred in the APC gene and leads to numerous colon polyps and colorectal cancer. Now let's briefly mention the hallmarks of cancer. We have a course dedicated to oncology that you can check out, where we go into all of these things in detail. But here we can just mention the key points. So we have sustained cell growth. We have another one in the hallmark of cancers. We have evading growth suppressor genes. We have resisting cell death, inducing blood vessel formation, enabling immortality, invasion, and metastasis. So cancer cells are like rule breakers. They grow uncontrollably, ignoring, for example, these stop signals and spread to other parts of the body. Some cancer risk factors could be genetic, so inherited mutations like the BRCA1 or 2 gene. Indeed, environmental, like UV radiation, tobacco, carcinogens in general. Lifestyle, diet, physical activity, alcohol consumption. They can be viral, so like HPV is linked to cervical cancer. Hepatitis B and C is linked to liver cancer. We have targeted cancer therapies. Now, what are these? These are drugs that are specifically targeting genetic mutations in cancer cells. For example, HER2 inhibitors. We have EGFR inhibitors. For lung cancer, we have PARP inhibitors for BRCA-mutated cancers. The advantage here is that it's fewer side effects compared to traditional chemotherapy. So targeted therapies are like very precise tools. They are precision tools that attack cancer cells without harming healthy ones as much. So the purpose for genetic testing for cancer could be, for example, to identify individuals at high risk for hereditary cancers. It can help guide personalized treatments. Tests, for example, for BRCA1 and 2 could give us information for breast and ovarian cancer, Lynch syndrome panel for colorectal cancer. And the implication of this is early screening and prevention strategies. And of course, family counseling for at-risk relatives. Now, cancer prevention and early detection. So prevention, you can do lifestyle changes like quitting smoking, healthy diet, exercising, vaccinations like HPV vaccine for cervical cancer prevention, avoiding environmental carcinogens. Another one is early detection, so regular screenings like having your mammograms done, colonoscopies, genetic testing for high-risk individuals. Challenges in cancer genetics. So it's quite complex because cancer is often caused by multiple mutations. Genetic testing and targeted therapies are actually quite costly and not widely available. And we always have to mention ethical issues like privacy of genetic data or discrimination risks. So the future of cancer genetics. Advances in genomics. So whole genome sequencing helps us identify rare mutations. Liquid biopsies detecting cancer through blood tests. Immunotherapy, so using the immune system to fight cancer. Artificial intelligence in cancer research, so predicting cancer risk and designing personalized treatments. Example, liquid biopsies are being developed right now to detect cancer earlier and less invasively. So in summary then, cancer results from genetic and epigenetic changes that disrupt normal cell growth. Key genes include oncogenes, tumor suppressor genes, and DNA repair genes. Genetic testing and targeted therapies improve diagnosis, prevention, and treatment. Future technologies like genomics and immunotherapy promise breakthroughs in cancer care. For in-depth detail into the world of cancer, please check our course on oncology. That's the end of chapter 12. Thank you for listening. Continue now to chapter 13.